Hello all, welcome to DNA Dialogue, where members of the genomics community in Australia and beyond can hear about new developments in health genomics internationally. I'm Tiff Bortwood, I'm the Managing Director of Australian Genomics. As we come together virtually today and in this National Reconciliation Week, now more than ever, we acknowledge the traditional custodians of country throughout Australia and their connections to land, sea and community. I pay my respects to Indigenous elders past and present and extend that respect to all First Peoples joining us today. I'm joining this webinar from Naarm, the lands of the Wurundjeri Woiwurrung people of the Kulin Nation. Please ask any questions using the Q&A function during the presentation. Select questions will be answered at the end during question time. And our speaker has also said that if there is a burning question that's relevant to the presentation at that time, I am allowed to interject to get your points across. If you're active on social media, we encourage you to hashtag DNA Dialogue and also please tag @Australian Genomics. So now it's my pleasure to introduce today's speaker, Dr. Leslie Biesecker. Leslie, you can turn your camera on and join me while I introduce you. Welcome. Leslie is a clinical and molecular geneticist and director of the Centre for Precision Health Research at the NHGRI. He uses genetic and genomic technologies to study the etiology of inherited disorders, of which his lab has elucidated many. He developed the ClinSeq program, co-directs a clinically certified molecular diagnostic lab within the NHGRI, is a past president of the American Society of Human Genetics and was elected to the National Academy of Medicine in 2016. So most in this audience would be familiar with the 5 variant classification categories used widely in today's clinical genetic and genomic testing, derived from the recommendations of the American College of the Medical Genetics and Genomics, the ACMG. The pathogenic, likely pathogenic, uncertain significance, likely benign and benign. So the ACMG is drafting what will be the 4th iteration of guidelines for the for variant classification framework, which will incorporate a number of recent advances, including a mathematical framework, incorporating objective quantitative assessments of evidence, and subdividing of the VUS category. So this is going to have significant implications for clinical diagnostic practice. And Leslie, we are eager to learn more. Thank you for joining us today.
Thank you so much for having me. It's a pleasure to be able to do this. Let me flip to my first slide, get it to presenter mode. Okay, looking good, Tiffany?
Thank you.
Wonderful. Great, thank you. Thank you for the lovely introduction, and pleasure to be with you here virtually. I would much rather be there in person. I love Australia, but we'll do fine with virtual today, so thank you for having me. So a little historical context here. Most folks who think about sequence interpretation or classification standards think about the paper that Richards et al. Published in 2015, and for those of you who do CNVs, the 2020 paper written by Aaron Rooney Riggs et al. But there is actually a larger context to this. Use that, which is this occurs in the context of 2 prior small nucleotide change recommendations. There's actually 2 Richards et al. Papers, which makes it confusing. There's Richards et al. 2008, and then 6 years before that, Haig, Kazazian, and co-authors actually penned the first set of recommendations. Now, if you try to find this Kazazian et al. Paper, you'll have a hard time because it isn't indexed in PubMed. If anybody's desperately interested in it, I'd be happy to send you the PDF, but but it's hard to find, and to be very honest with you, it isn't terribly useful. It might be more interesting as a historical, almost amusement exercise for you, because those of you who do variant classifications might get a bit of a chuckle out of how people were talking and thinking about variant classification 22 years ago. It's pretty interesting. But we're building off of the 2015 Richards et al. Effort, and because of the confusion with the existence of the Kazazian paper, the two Richards et al. Paper, we just decided to switch to a versioning nomenclature. So we call them V1, V2, V3, and we're working on V4 now. And so we're applying that retrospectively. And then what we have tried to do with V3 and the CNV recommendations was begin to harmonize terms as well as align the methodologies. So the CNV guidelines attempted to incorporate some of the points-based approach that we are going to be piloting in SVC4, and the CNV group has committed to completely redoing their CNV recommendations after V4 of the SVC comes out and make it fully Bayesian so that it will be completely compatible, which is really important, right? Because, for example, you can readily think of disorders where an individual might be a compound heterozygote for a small nucleotide change and a CNV, and you want to be able to classify those and write a report in an integrated way so that they both make sense at the same time. And to that end, they have to be compatible with each other. So it's very important to have these things aligned and harmonized. Now, we've also taken the attitude that some of you may experience, and one of the most common questions we get is, when the heck is version 4 going to come out? I hope it will be later this calendar year, but it may not quite be that soon. And to be honest with you, I think 9 years is too long between V3 and V4. And so we have adopted the approach going forward that the ACMG secondary findings work group has piloted, which is they're going to be making major recommendations and revisions infrequently, and those will be the integer versions, and then they will do decimal versions of version 4.1, 4.2, etc., when they want to make minor tweaks or correct issues in a timely way so that you don't have to wait a long time between the recommendations. And so you can anticipate that these big events will come in the 5 to 10 year window frames, but then smaller revisions will come out more frequently than that. I'm the co-chair. I have a wonderful co-chair working with me, Stephen Harrison, who used to be at the Broad Institute, is now at Ambry Diagnostic Labs, with a whole host of great people working with us to push this forward, mostly from the US, but with solid international representation. And I do always want to call out Heidi Rehm to my right in this photograph. Who is really one of the stars of clinical genetic testing, genomic testing. Heidi's really a pioneer, a leader, and one of the most thoughtful people I've ever had the pleasure to work with. And just a hot tip, if anyone ever gets a chance to collaborate with Heidi, take that opportunity. You'll be repaid in spades. Okay, so what are we doing with 4.0? So the formal charge here is to outline rules to determine the actual likelihood that a variant in a gene that has an established role in a monogenic disorder is pathogenic for that disorder. Now, this more formal framing of what we're setting out to do wasn't actually explicit in SVC3. But we are making it more formally explicit in order to underpin the mathematics that we're setting up here. And so conceptually, it continues with the scope of SVCv3, but we're making it more formal, and we're really pushing hard on this notion of convincing and reminding people that this is a probabilistic enterprise, and what we are trying to assess is the likelihood of pathogenicity. Now, this other qualification here that I put in bold, a gene with an established role in a monogenic disorder, excludes two things. So two things that we're not doing is we're not going to be making statements about the pathogenicity of variants if the gene has not been established to have a role in the monogenic disorder. That is, these rules are not necessarily appropriate for establishing the relationship in novel genes with phenotypes. Nor is it appropriate to use these rules for establishing the role of variants within disorders that are polygenic or multigenic. That's a separate enterprise worked on by a different group, and be happy to engage in a discussion about what is a monogenic disorder. Not an easy question, but I think most of us have an intuitive feel for what it is and isn't, and you want to be sure that you're operating within those bounds when you're doing this work. So we've actually formalized this a bit and nailed this down such that if you look on the left column is the validity of the gene phenotype or gene disease relationship. That is, if there's a definitive or strong relationship of pathogenic variants in that gene to the disease, then you can make the full range of assertions about variants within that gene in your clinical testing report. But if that relationship is only of moderate validity, then we are going to say that you cannot make an assertion of pathogenicity. Because, of course, if the likelihood of the gene disease validity is only moderate, you can't really be saying that the relationship of a variant within that, a single variant within that gene is of more than moderate strength of relationship to a phenotype. That makes sense, right? And if that relationship is limited, you cannot make an assertion higher than VUS. If it's less than limited, then we recommend a slightly different terminology. We recommend that one does not even begin to classify the pathogenicity of a variant in this 5-step PLP-VUS-LBB range, but just say only that you are reporting on a variant in a gene of uncertain significance, and do not make an explicit statement about the variant itself. If it's lower than that, you really shouldn't be writing anything about it in a report because you just don't have much to say about it. Okay, so the applicability, which again is conceptually similar to the scope of V3, is that these recommendations will be applicable to variants in all genes that are associated with monogenic disorders, whether that variant was identified by a single gene test, a multi-gene panel, or exome or genome sequencing. And so it's intended to cover a wide range of testing scenarios, even though that leads to some statistical or probabilistic challenges down the road. We can talk about that in the Q&A if you wish to pursue that. But we're taking a unified approach here. And we do acknowledge, and this is this concept with the versioning, that our classification approach has to evolve as the technology evolves and as our knowledge of these disorders and the mechanisms of disease for these variants evolves. One of the big changes we're making, which really didn't exist at all in V3, is to begin to shift variant classification from professional judgment to quantitative evidence whenever that is feasible. And Richards et al. Did not make much use of that. Richards et al. Was purely an expression of professional judgment, sort of the art of variant classification, if you will. And they made no bones about that, and I don't think anyone should feel bad or be critical of V3 about that, because they did our field a huge service. Just the process of putting down in black and white what criteria labs were considering and relative weighting of those criteria and a way to combine criteria, even though it was professional judgment, was a huge advance over V2. If you read V2, again, you'll be pretty disappointed in how vague and non-specific all the recommendations are in that. So Richards et al. Did a superb job advancing our field. Another terminology change we're switching to is the term classification. So we are talking here in these recommendations about variant classification, and we are distinguishing that from what we're calling clinical interpretation. So the former is the process that a laboratory geneticist performs when they aggregate the evidence, all available evidence about a variant, to assess the likelihood, probability again, that a variant is causally associated with a given Mendelian or monogenic disorder. So that's what the laboratory is doing, and that's what they are putting on the clinical testing report. In contrast, then the next step is clinical interpretation. And that's the process of assessing if a given variant of a given pathogenicity is indeed associated with disease or the risk of disease in an individual patient. So this is what the clinician is doing in the clinic after the laboratory test report is received, and they are deciding the relevance of that test report for the patient that they are caring for. And we thought it was important to distinguish those because they're different probabilistic activities, and it's important to be clear when you're doing one versus the other. And it's also be clear, something else we could discuss if we have time, about we have to be a little bit careful about some circularities that one wants to avoid when doing variant classification. And the key takeaway from this is we're not just arguing about the words people use. It's very, very important to recognize that the likelihood of the pathogenicity of the variant is not equal to the likelihood of disease in the testee. Critical variance. So if I say a variant is 90% likely to be pathogenic, I will call it likely pathogenic. And if that variant came from a patient, it is not correct to say that that patient has a 90% likelihood of disease. That depends on the likelihood they have of having that disorder before you did the test, exactly as is the case for every other medical test in every other disease state. And we think here about three different concepts. There are different things that one thinks about when one has a test report in a patient. There is the clinical finding, or sometimes a clinical phenotypic diagnosis. That is, I may say, my patient has 6 features of Marfan syndrome, and therefore I say that based on the clinical findings, I think the patient has a clinical diagnosis of Marfan syndrome. Perfectly legitimate medical decision to make. Now I take a sample from that, send it to the laboratory, and the laboratory comes back with a molecular finding. Again, this probability, this probabilistic assessment, their genotype, and they say they have a cysteine 570 arginine missense variant, and I think that that is a likely pathogenic or pathogenic variant. And then I take that test report and I then bring it back to the clinic, And then I say, I'm now going to make a clinical molecular diagnosis, which is this notion of a dyadic assessment, two different attributes, clinical and molecular. I'm putting them together. And then I say, my patient has FBN1 related Marfan syndrome. Those are three different assertions to make. They have different implications, different mathematical probabilities, and they're important to keep separate. Okay, so what we're doing with the SVC v4 is to revise the current standards, and what we want to do is readdress the appropriateness of the individual criteria. We looked at all the criteria that were used in v3, reevaluated them. Search out for ambiguities in the specification and the application of those criteria, of which, and all of us will agree who work with V3, there were plenty of them in V3. V3, at least in the States, we kind of analogize it to constitutional law, which is a bunch of people sitting in a room arguing about what the Constitution allows and doesn't allow, or outlaws or doesn't outlaw. We have to argue about how those V3 criteria do or don't apply to a given variant. Some of that's art, some of that's science. Then we have to assess the strength of each criterion individually and in combination, and how to combine them. And one doesn't always just add them together, because logically that is not justifiable. So you have to be very thoughtful about that. And then think about what to do about the lack of guidance when benign and pathogenic criteria are applied together. V3 stated that if you have evidence for benignity and pathogenicity for a variant, that it's a VUS. That was too simplistic. We all know that there's plenty of situations where you might have a huge amount of evidence for benignity and a little bit of evidence for pathogenicity. That's not really a VUS. That's going to be a benign or a likely benign classification. So V3 didn't really have a mechanism for weighting that when you have both kinds of evidence. And again, as I mentioned earlier, whenever possible, what we wanted to do is take the V3 criteria and transform them into quantitative parameters and assess objective quantitative data that can be used to actually weight the pathogenicity of those variants so that it's externally valid, not just internally consistent. So I'm going to walk through a couple things we're working on just as examples of things that are going to be changing, the structure of what we're doing, the evidence codes that we're going to be using, and show you some of the specific tables and flow charts. Now, I put in red here, draft. Please, please, please don't take these as givens. They could change before the final recommendations are published, but the generalities will be helpful to you. Okay, in comparing V3 versus V4, what you'll recognize is that both versions of these recommendations consider multiple individual lines of evidence and then have a combining method to arrive at the final classification of a variant. The lines of the evidence from V3, you will recognize almost all of them persist into V4. Again, V3 was a good paper. They really did a service for our community, and they considered nearly all of the relevant concepts. Created a few new evidence types, but mostly they are carried forward from the prior one. The structure for how that evidence is considered individually, as well as how we aggregate them, is different, so you will notice some changes there. Now here's our favorite old V3 codes, and I don't think anybody in clinical genomics likes these codes. None of us can remember them. To try and remember which one is PS3 and which one is PS4, I'll be darned if I can remember them. You probably can't either. There were a lot of problems. Sometimes the same evidence concept was represented in multiple places with different labels. So here's an example of the de novo criterion when you had confirmation of parentage. That was a strong evidence in V3. And in V3, if you didn't have confirmation of biologic parentage, it was a moderate strength criterion. But the first one was called PS2, and the second one was called PM6. And again, nobody could remember those. So it wasn't a naming system that anybody liked. For some of them, the default was a single strength for many of the codes. And we all know that, in fact, there are nuances to almost all of these evidence types. And this is a great example, the functional evidence code, PS3. Not all functional assays have the same strength. V3 aggregated that and said in most cases a robust assay, especially one that was used in a clinical setting, is probably strong. It wasn't unreasonable, but again, you want to have a little more flexibility so that you can calibrate your evidence and use it in whatever strength it comes. This is going to change enormously during the lifetime of V4 with the development, as you know, of the multiplex assays of variant effect. These are huge data sets that are being generated for a number of genes, and there's going to be a wide range of readouts of those multiplex assays at varying strengths, and you'll want to be able to apply those at the proper strength for each output of each variable. And let's see, continue. I already said that, so we can skip that. Okay, so some of you may be familiar with a paper Sean, Tev, Tegan, and I piloted a few years back and then adapted to a little more of a simple approach where we looked at Richards et al. V3. And what we recognized in V3 was that it was implicitly Bayesian probabilistic mathematics. Now, what's really interesting about this is I've talked to members of the V3 committee, the Richards et al. Committee, and I have been assured by all participants that I've talked to that the word Bayes was never raised in any of their discussions. And I think that's a super interesting observation because it's a really powerful endorsement of how fundamental Bayesian probabilistic thinking is to diagnostic testing. They were doing it. That's what they had in the back of their mind, and their recommendations were immediately and readily adaptable to a formal mathematical equation that uses Bayesian logic, but they never thought that they were doing that. Again, it's implicitly Bayesian. And so we recognized the Bayesian nature of that. And then a few years later, we created this points system where you could then take each piece of evidence, and on the left of that table is the V3 evidence strengths, and you could quantitate them, and we just back calculated the odds path, which is the conditional probability in a Bayesian probabilistic assessment, to yield the output that matched the V3 et al. Recommendations of the 1%, 10%, 90, and 99%. And those odds path with the combining criteria in nearly all examples of the V3 combining table led to the same conclusion. Nobody liked working with those unusual numbers, those odds path numbers, and so the points adaptation allows you to just do 1, 2, 4, 8 geometric scaling of the points, and then you can just add them together. You can add the positives and the negatives together, and it gets you a very simple output. You can just do that right in your head for each criterion. Now, a big change as we are going to propose a subdivision, as Tiffany mentioned in the introduction, of VUSs. We recognize that there's a lot of challenge, and it's especially significant challenge in the boundary between the high end of VUS and the low end of likely pathogenic. It makes a lot of people very uncomfortable when they're working with a variant that is close to that boundary and trying to decide whether it's a VUS or LP variant. And so what we decided to do is to make that more explicit so that we can make labs more comfortable with those high VUSs and reporting them as high VUSs, instead of artificially trying to push them to LP because of concerns that they had some sort of a gut feeling that they were actually more likely to be pathogenic, pushing them in the absence of evidence into that LP category. So the point scale fits itself naturally to this distribution. A lot of debate about whether these three windows should be symmetric as they are here, 2 points to each window, or asymmetric with a wider gap in the middle, but we've tentatively settled on the 3 equal intervals. And recommend that for now, labs won't be required or expected to initially report these VUS subcategories, but we recommend that all labs begin to track the criteria that they're using for the classifications using the point system. Keep that in their internal systems, get used to that tracking of those points and see how variants would fall. And then as labs get more comfortable with that, they can begin to implement the VUS subclassification. Now, the utility of these subdivisions is probably most important with the VUS high category. Now, our group does not have the charge of making recommendations about what should be reported to a patient. We only make recommendations about what should be in the report. And so we're going to recommend that the VUS high begin to be used when labs are comfortable with it. But I think, and as you will all recognize, in diagnostic testing, there are a number of situations where a VUS high variant classification could be highly relevant to a clinical molecular diagnosis, again, in a diagnostic testing situation. They will almost certainly not be useful in a screening or secondary findings situation. The posterior probability of disease with even a high VUS in a screening diagnostic setting, and this was what I was hinting at earlier about when I said, remember, the classification pathogenicity of the variant is not equal to the likelihood the patient has the disease. When you're in a screening or secondary findings setting, the prior probability of disease in the patient is low, probably close to the general population incidence of the disease. So 1 in 400 for hereditary breast and ovarian cancer, let's say, in most testing scenarios, 1 in 500 for hypertrophic cardiomyopathy, much less for other rare disorders, right? And so when you have that low prior probability of disease, finding a VUS in a patient has a terrible posterior probability of identifying disease. If you're in a diagnostic setting, especially in the setting where you're testing a patient that you think clinically has a pretty high likelihood of having the disorder before you do the test, i.e. a person with early onset cancer that has a strong family history of disease, that's a high prior probability of disease, right? If you find in that patient only one variant and it has a VUS high classification and you don't find LP or P variants, that VUS high variant has a pretty darn good likelihood of being the explanation for that patient's disease, and the fact that it's VUS high is a signal to the ordering clinician, that they should work hard on that case to see if there's additional data or information they can generate from that proband or their family that might push the variant classification even higher. And that's the kind of interplay that we want to facilitate between testing labs and ordering clinicians to get families to a proper diagnosis. Okay, so the overall structure of the evidence, we've broken it into more memorable categories. So we broke it up into two high-level categories, which is human observational data, and then the second one is functional and predictive data. And the human observational data include things like counting observations, observations of the variant either in people with disease or people who don't have the disease, as well as locus data, that is segregation and the specificity of the phenotype in a tested individual. So those are the human observational data types. And then we tried to name them with abbreviations that more match the words that one uses. So the observation counting, you can see there, is OBS _AFF. That's observations in affected individuals, or observations in unaffected individuals, okay? Or observations in the general population. And so you can use those three categories, and they're much more memorable than those old codes. And then we've completely dropped the notion of using the S supporting M moderate. I'm sorry, see, even I screwed that up. P was supporting, M was moderate, S was strong, and V was very strong. Those were the strength codes in V3. Now we're just using the points. 1 is supporting, 2 is moderate, 4 is strong, and 8 is very strong. And then it also allows us to generate much higher levels of strength when the data support that. So it's just OBS, AFF, let's say +4. And then you know it's observed observations in affected cases, and it gives you 4 points of pathogenic evidence, and it's right there in the code. Okay, so again, these labels are all draft, and I'm sorry, did my speaking a little bit out of order with my slides. Again, the codes match the descriptions, and they have no inherent strength. It's just the point value that's appended to the end of the code. Okay, and as you may remember from V3, most of V3 was prose. It was written out descriptions of what the evidence was. And instead of that, we're switching in V4 to more logical decision trees whenever we can. Now, this has risk, and we have some trepidation about this. We think it's the right thing to do, but it's critical that all users understand that the decision trees that we will specify encompass some of the more common combinations of evidence that you will encounter. But the most critical thing is to recognize that no decision tree can incorporate all possible scenarios. We cannot do that. And so just like in V3, an expert, a knowledgeable person has to look at the application of the evidence, use your knowledge of genetics and biology to correctly apply the criteria from the decision trees and use that to classify a variant. And I can tell you, you can see in the bottom here, that's meant to be a little humorous, this is not draft. I guarantee you this caveat is going to be in the paper, and it will apply to everything that we specify. Now, the way we generally have structured these decision trees is moving from left to right, is to think about, start with whatever type of variant it is, look at the predictive data that one has for that type of variant or that specific variant, then assess the exon and transcript relevance of the variant, which is to say, is that variant in an exon that is present in all clinically relevant transcripts. If it isn't, then you should either not use that criterion, or you should lower the strength of evidence you're going to apply. Then you can, in some cases, assess the importance of that region of the predicted protein, its impact on some known function of that protein product, and then take what you have, which is mostly predictive there, and then look to see if there are other similar variants that have relevance to the pathogenicity of your variant. And you might remember that from the V3 recommendations, where it used to be a strong piece of evidence, for example, if it was the same amino acid change being caused by a different cDNA change, that was a piece of evidence supporting pathogenicity if that other same amino acid change was also judged as pathogenic. This going back and forth between the variants, it gets confusing, and so we created this nomenclature of VBC. That is the variant being classified. That's the variant that came out of the patient who's being sequenced that you're working on. And we use variant being classified throughout the document, and then we refer to the other variants as informative variants, so you can keep them separate. So here's an example of a decision tree for a nonsense variant. Now, this looks kind of different from V3, but if you walk through some of these variants as they're released, some of these trees, as they're released, you will actually recognize that the logic, for example, of this nonsense variant tree is pretty darn similar to the tree that was promulgated from V3, Richards et al. So you look at, number one, it's the type of variant. It's a nonsense variant. You see whether it is upstream or downstream, positioned within the predicted protein, and what its relationship is to other potential in-frame start codons, for example, if it's at the 5' end. And then you look at things like exon or transcript relevance to see if it's in an exon that's present in all clinically relevant isoforms of the mRNA. And then you look at your informative variants. And so, for example, if you have a pathogenic or likely pathogenic variant that is also predicted to lead to nonsense-mediated decay that's in the same exon as your variant, that adds evidence. And the more of them that there are, the more evidence that it adds. So you can follow it across here. Nonsense variant. Located prior to 50 nucleotides, its relationship to other methionines, and then that leads to a code, and it's a code, a null code, because it's a nonsense-mediated mechanism of disease. And this is a decision tree. This is an important caveat. It's not shown on this flow diagram. This is a decision tree that should only be used if loss of function is a mechanism of disease of the gene phenotype dyad that the variant being considered is being evaluated for. So I said this earlier, so I can skip that. And so if you follow this through here, if it has this attribute, but it's located more than 50 nucleotides upstream, and the predicted loss of function variant being considered is not upstream, that's why there's a no here, of an alternate in-frame methionine, and it's in an exon where the transcript has that exon in all relevant isoforms, you're starting here with plus 7 points. So just this criterion alone is already getting you 7 points, which, if there were no other criteria relevant to this variant, would already be taking you into the likely pathogenic range. If you had similar pathogenic variants, you can increase those points, and again, you can see the scaling here. And we also set it up so that if there are similar variants that have a benign classification, you would actually lower the points. So you would add negative points to other benign variants that are in the same similar region of the protein. And then the total output here can range from -4 to +10 points. So if you had it was present in all transcripts, and let's say you had one pathogenic and one likely pathogenic variant in the same region and no benigns, that would be 3 more points, and that would get you to +10, which would be at the margin of pathogenic. Now, if you're looking at a nonsense variant when nonsense-mediated decay is not predicted to occur from that, we capped it at a lower limit of the maximum amount of points you can get. And then you also have to look at this thing called region information. That is, and this is similar again to the concept that was present in V3. If your variant removes or alters a critical functional domain, sorry, the slide slipped there, that's relevant to the disease mechanism or removes more than 50% of the protein, even if it's not causing nonsense-mediated decay, you can add 4 points. But your starting point here is 3 because, again, if nonsense-mediated decay is not predicted, you should start at a lower point than if nonsense-mediated decay is predicted to emanate from that variant. But you can add more points if it's a critical region, and you can add more points if there are informative variants that are also pathogenic or likely pathogenic. Our missense variant decision tree is a different structural tree in that it has 2 parallel pathways, both of which should be evaluated for every variant. And this is a recognition of something. This wasn't really in V3 Richards at all, but as you all know, a variant that appears to be a missense variant can instead actually have a splicing effect rather than an effect on the amino acid sequence alone of the protein. And so we decided we should have a parallel decision tree where you look at the amino acid effect and you look at the potential splicing effect, and when you get to the right end of this branch tree, you look at the scoring for the protein change versus the splicing change, and you select the evidence that is more positive. This is based on the recognition that in silico tools can have actually significantly higher strength of evidence than Richards et al. V3 allowed. V3 only allowed supporting pathogenic evidence for in silico predictors, and it didn't do a very good job, just to be honest with you, about how to use, which predictors to use and how to use them. We did this paper in ClinGen where we calibrated a number of tools a very large set of nearly 12,000 variants and could calibrate these tools and actually measure the strength of evidence that they had. And happy to report, some of these tools work very well, much better than supporting strength of evidence with many variants when you calibrate them, and you can actually justify using up to strong evidence +4 points or -4 points for some of these meta predictors. Again, if this were the only evidence you had for a variant, the most you could get out of it is a VUS high. So no one should worry too much about these predictors overcalling variants. I think that's a very unlikely problem, but it's definitely better than supporting. Okay, so first you assess the prediction change from one of these meta predictors, and then you assess the exon and transcript relevance. And here the exon transcript relevance is handled a little differently. Instead of adding points as it did you saw in that prior example, what we are doing here is basically adding, if you will, a discount. So if the amino acid change that you're looking at is present in only most of the transcript or isoforms, then you take whatever score you have from the meta predictor and multiply it by 0.5. If it's not expressed in clinically relevant transcripts, you shouldn't be using the meta predictor at all because it's just not relevant. And then again, we use the similar amino acid changes, which is similar to the criteria that were present for the informative variants in Richards et al., quite analogous. Then I don't have slides on this, but we also use in silico tools to predict the splicing impact. The main one we have the most experience with and we'll give examples for, although we're not specifically endorsing it, is Splice AI, and that can generate pretty robust scores for some variants, and you'll see examples where you can get a more positive score out of the Splice AI, splice consequence pathway than you can out of the amino acid change pathway, and that's telling you that that variant is more likely to have a splicogenic effect than it is to generate a stable protein with an amino acid difference. So the most common question we get about SVC4 is, when is it coming out? We hope to be piloting it soon. The steps are that we have to submit the draft standards to the ACMG Molecular Laboratory Quality Assurance Committee. They review that, they will review it and then submit it to the board. The ACMG posts, as you know, recommendations to all members for comment, and then the draft will be modified based on that feedback. The most important thing you can participate in, if you're interested, is in piloting. So when we're ready to release the recommendations at this first step, we will release draft flow diagrams and guidelines, and you can begin to try them on your variants and give us feedback on that. So I'll stop there. I know I went through that pretty quickly and gave you a few examples. Hopefully it gives you a flavor for where we're going with the new recommendations, and I'll be more than happy to answer any questions.
Leslie, thank you so much. That was such an informative presentation, and acknowledging the draft status on all aspects, apart from the fact that people still need to use their knowledge of genetics and biology to correctly classify variants. Now we've got lots of questions in the chat, and
Great.
We'll get to as many as we can. I'm not surprised given the attendees who are with us today. There's lots of people these new classifications will affect. So Eric asks, does V4 account for the same variant having different classifications for different phenotypes associated with that gene? Will gene-specific classification guidance from ClinGen be updated to the V4 framework?
Brilliant question, Eric. Yeah, this is one of my pet issues. What we're moving to, I talked a little bit about that dyadic structure for describing a patient. ClinGen is also moving to a dyadic disease designation structure for designating disease entities. So therefore, one that we work on, which I'm very familiar with, is RYR1. You may know RYR1 is associated with malignant hyperthermia. It's also associated with several myopathies. The mechanism of disease is distinct, even though it's the same gene. There are distinct mechanisms of disease, and the rules for one must be different from the rules for the other. Therefore, the laboratory should be evaluating the variant for both of those entities, and we will be considering those to be two separate disease entities for the purpose of variant classification. That is, RYR1 related centronuclear myopathy is one entity. RYR1 related malignant hyperthermia is a separate entity. They should have separate rules, and the variant classification could be legitimately P for one and VUS for another. And that's actually not a conflict. Those should be two separate freestanding classifications, both valid. And we need a way to specify that. I will tell you, we are struggling right now because ClinVar cannot accommodate that. And we're working hard with NCBI to get them to recognize the importance of distinguishing these entities so we can do exactly what you're asking.
Excellent, thank you. So Chirag, thank you for the talk. Also is interested whether you've assessed V4 unlikely pathogenic variants reported within V3 and found that some have been downgraded. So I was also interested in this. What are the implications for reclassification, particularly in the context of reanalysis, for example?
It's a great question. In fact, LPs, well, pathogenic classifications so far, we have done only because again, the recommendations aren't final, so I can't really answer your question. It's a great question. But to the extent that we're able to start testing them out, what we're seeing is pathogenics don't move much at all. A decent number of LPs shift up to P, and a modest number of VUSs shift to LP, and a large number of VUSs shift to LB. And I think what that is a recognition of is that we as a profession internationally are over-classifying things a bit. And I think I don't know what it's like in Australia. In the States, at least, we have a rather terrible problem with medical legal liability and lawyers and lawsuits and all that rubbish. And so labs are often nervous about assigning a variant, a classification of LB, when there is a small amount of benign information. So they classify them as VUS because they think it's safer. That is a systematic overestimation of the pathogenicity of variants. And I think with the criteria being more explicit and quantitative, it's going to support people being more comfortable, I hope, going with LB. And a great example of that is for many missense variants, sometimes a novel missense variant, the only piece of evidence you have is the in silico predictor. And a lot of labs say they're nervous about downgrading a variant to LB based solely on an in silico predictor, but the data support it. And so you should be doing that. And if you think about the likelihoods, especially if you're doing exome or genome testing, right? Think about it, right? In Richards et al., the default of a variant for which you have no evidence is VUS, which means pathogenicity is between 10 and 90%, right? That's what VUS means. If I sequence an average patient, I'm going to come up with an exome 30 to 40,000 variants. Do you really think that more than 10% of those variants are actually disease causing for a Mendelian disorder? That that person harbors 3,000 to 5,000 pathogenic variants? No way on earth is that true. It's mathematically ridiculous. That's the way we have to start thinking about this, is this more formal probabilistic method, and we will push those variants down back where they belong.
Excellent, thank you. Now, Francesca makes more of an observation. It'll be interesting to consider whether the scoring system for VUS high is the same in a prenatal and postnatal setting, given the phenotype evidence strength is not the same in both, and cites Tony Rossioli's work in that context.
So this gets into a little bit of that circularity issue that I was talking about. The phenotype of the testee, which used to be the PP4 criterion in V3, can inform the pathogenicity of the variant, but one has to be very careful when one is doing that, because you are biasing that. And as my friend, the population geneticist Alec Wilson, used to tell me, Les, you have to remember that every variant that the patient harbors is associated with their phenotype. It's like, oh, right, of course it is. So we have to be really careful when we do that. And so it's a tricky exercise, and it's going to take us all getting a little bit used to this. When you're doing a screening setting or a setting where you don't have phenotype information, you don't have that, you don't have as much to go on to start your classification using that concept. Whereas the patient with early onset cancer and a terrible family history, you're starting with a lot. And we have to understand how we properly use that to classify the variant and then not double count it when we're saying, oh yes, now I'm sure the patient has that disease, because there's a circularity there. And we have to be very careful to break that, and we'll work on doing that so that we can do both those things properly. It's a great question.
All right, we have many questions that we're not going to get to, I'm afraid, because we are at time. Sorry, everybody who asked questions. But look, I think it's just a testament to the degree of interest in your talk. It's been an absolutely fascinating presentation, Les. Thank you so much.
Thank you for having me. It's a pleasure.
A delight.
Stay well, everyone.
Thank you. And we hope to see you in Australia sometime soon.
Terry, take care.
And thank you all for joining us. Our next DNA Dialogue seminar will be held on Thursday, the 27th of June, and will feature Dr. Anna Topp from the UK's Newcastle University talking about the Latin-seq project. I hope you'll be able to join us then. Thanks very much.